Welcome to the Longevity Papers podcast. Our mission here is pretty straightforward. We cut through the huge amount of scientific literature coming out every week.
Yeah, articles, preprints, sometimes even research notes people share.
Exactly. And we pull out the really important nuggets of knowledge, the insights specifically for you, the biotech researcher working in this field.
Think of us as, well, your shortcut,
Yeah.
helping you stay informed about what's genuinely moving the needle in longevity science.
We try to give you the signal, with a healthy dose of critical skepticism thrown in.
And this week, looking at July 21st to July 27th, 2025. Well, there's certainly been a lot published, a real wealth of studies. But as always, our goal is truth-seeking. So we need to manage expectations right up front. We're going to be pretty candid about what we found or maybe what we didn't find.
Absolutely. We went through, 53 papers this week? All from credible institutions.
That's right.
And our focus, always, is finding work that makes you stop and think. Something that genuinely demands a model change in how we approach longevity.
Or research, or even how we design interventions. We're looking for that truly surprising stuff.
Yeah, the paradigm shifts.
And being totally honest. While a lot of the papers were interesting, well-done science, maybe 95% felt largely incremental this week.
Right.
We've picked out the top 4 that we think warrant discussion, maybe for their novelty or some mechanistic insight. But, and this is important, none of them, in our view, present that kind of transformative breakthrough.
Nothing that screams add a decade to human lifespan this week.
Not this week, no. And we'll get into why as we discuss each one.
And that's exactly why we do this. We apply that critical filter, right? Especially looking out for findings that might just be, statistical luck. We'll talk about what previous research this builds on and, importantly, suggest the next experiments we'd want to see.
Trying to give you enough detail to really get the analysis without having to read every single figure yourself.
Yep. And just a quick note for everyone listening, when we mention bioRxiv, we'll be pronouncing it bioRxiv.
Good point. BIORXIV, bioRxiv.
Okay, let's jump in. First paper up, Large Language Model-Based Biological Age Prediction by Lai and colleagues.
Right. This one's out of Tsinghua University, published in Nature Medicine just last Thursday, July 24th.
And what's striking here isn't just, another aging clock, it's the scale. And using an LLM this way.
Totally. It's the first framework like this predicting organ-specific biological age. That's the new angle.
The data set is huge.
Oh, massive. Health reports from over 10 million participants.
Yeah.
That's pretty unprecedented for this kind of work.
So what did they find? Did it work?
It did, surprisingly well in some ways. The key finding they're highlighting is that it actually outperforms established epigenetic clocks like Grimmage in predicting mortality.
Really? By how much?
They reported a C-index of 0.757. Compare that to Grimmage, which is often around 0.65. So that's a notable jump in predictive power.
Okay, that's interesting.
And the age gap, the difference between predicted biological age and chronological age that the LLM calculated, it was strongly associated with a whole range of aging-related phenotypes and health outcomes.
So it seems to correlate well with actual health status.
Exactly, which suggests it has real potential for personalized aging assessment, maybe even in large populations, moving beyond just your birth date.
Okay, powerful prediction. But here's the question for us, for biotech researchers. What's the mechanism? What's the catch?
Ah, yes, the caveats. And they're significant.
Always are.
The main one, it's a black box LLM. Inherently, it lacks mechanistic insight. We see these strong correlations, fantastic for prediction, but
Correlation isn't causation, the classic problem.
precisely. And crucially, there's zero validation yet on whether these predicted age gaps actually respond to known longevity interventions.
Right. Can you actually change this LLM predicted age with something like rapamycin or metformin?
We just don't know. Is it tracking a fundamental aging process that's malleable, or is it just a very, very sophisticated prognostic tool picking up on subtle health declines? Big difference.
So if you're in biotech, you see this tool. How do you take it from prediction to something useful for developing interventions? What's needed?
Well, first things first. Intervention studies. You need to test if this LLM age can be reversed or at least slowed by things we think work.
Like in primate models, maybe.
Primate models would be ideal, yes,
Yeah.
or even rigorous human trials, though that's longer term. Also, critically, you'd want to compare its predictions directly against established clinical frailty metrics.
To see if it's actually adding new information beyond what doctors can already assess.
Exactly. Does it give us a genuinely new clinical signal or just repackage existing ones?
So putting it in context, It builds on Horvath's clocks, obviously.
Definitely. It's standing on the shoulders of epigenetic clocks and other biomarker clocks like PhenoH. But the innovation here isn't really biological discovery.
It's technological.
It's a technological leap, yeah. Shifting towards AI for clinical utility for large-scale population health assessment.
So for biotech, maybe the value is as a faster endpoint in big trials.
Potentially. It could accelerate things if it proves reliable and responsive. It's a step forward in assessment technology, not necessarily aging biology itself this week.
Okay, let's move to the second paper. Still on biological age, but a totally different approach. DNA Methylation Aging Atlas across 17 human tissues by Jacques et al.
Right. This one's a preprint up on bioRxiv from Monash University, posted Thursday, July 24th.
An atlas, 17 tissues. Sounds incredibly comprehensive.
It is. It's a meta-analysis pulling together data from over 15,000 human methylomes. Just a huge data set covering a wide range of human tissues.
So what did they pull out of that mountain of data? What are the key insights?
They identified a gene, PCDHGA1, that's PCDHGA1, as what they call a conserved cross-tissue aging driver. That's potentially quite significant if it holds up.
A single gene driving aging across many tissues.
That's the suggestion. They also highlighted NAD plus metabolism as a resilient module in aging, something that seems to hold up relatively well across different tissues as we age.
Interesting. NAD plus comes up again.
It does. And they linked epigenetic entropy, basically, the randomness or disorder in methylation patterns, to the fragility of methylation networks. And they identified specific disruptors, genes like MEIS, HDAC4, and some HOX genes that seem to make aging signals worse.
Okay, so it's providing a really detailed map of epigenetic changes.
A very detailed map, yes. It could offer a roadmap for finding new therapeutic targets, maybe things that work across multiple tissues, encouraging people to look beyond the usual suspects.
Sounds potentially huge, but you flagged it as incremental earlier, so why isn't this a model change yet? What's missing?
The big missing piece is functional validation. It's an amazing descriptive atlas, incredibly valuable for generating hypotheses. But those correlations, those identified drivers, they haven't been functionally tested yet.
So we don't know if tweaking PCDHG1 actually does anything to aging.
Exactly. And the link to NAD plus metabolism, while interesting and fits with other work, it's largely building on what people like David Sinclair and others have been saying for years. It confirms it in this large data set, but it's not fundamentally new ground in that specific area.
It maps the territory, but doesn't yet show us how to reshape it.
Well put. We need to see if manipulating these targets actually changes the aging trajectory in a functional system.
So for a biotech researcher wanting to build on this, what are the obvious next experiments?
CRISPR knockout of PCDHGA1. That's the big one. See what happens in organoids, or even better, in vivo models. Does knocking it out slow or reverse aging markers?
And for the NAD plus part?
Direct NAD plus precursor trials, maybe specifically in aged tissue models derived from this atlas data. See if boosting NAD plus really shores up that resilient module they identified. Got to move from observation to intervention.
Okay. And how does this atlas fit into the bigger picture of epigenetic aging research?
It definitely expands significantly on previous work, like the multi-tissue maps from the GTEdx project. It gives us much finer resolution, much more detail across tissues.
Better clocks? Maybe more tissue-specific clocks?
Potentially, yes. But fundamentally, it still reinforces our existing understanding. Epigenetics changes profoundly with age. It gets noisier. Certain pathways are involved. It deepens that understanding, provides new targets, but doesn't radically rewrite the core concepts of epigenetic aging.
Got it. Deepening, not pivoting. Okay, let's pivot ourselves now. Hot area. Senolytics. Paper 3. GLS-1 inhibitor as a senolytic agent in vivo by Okamura et al.
Yep, this one's from the University of Tokyo. Also on bioRxiv, posted Wednesday, July 23rd.
Senolytics, clearing out those senescent cells. Always exciting potential. What did this group find? What's GLS-1?
So GLS-1 is glutaminase 1, an enzyme involved in glutamine metabolism. They show that PPTES, an inhibitor of GLS-1, selectively clears certain types of senescent cells in aged mice.
Which types?
Specifically, inflammatory senescent fibroblasts and endothelial cells. And they dug into the mechanism.
How does it work?
It seems to disrupt glutamine metabolism in these senescent cells. This leads to reduced interferon signaling, IFN signaling, and interestingly, an increase in protective T cells, which might help clear the remaining senescent cells.
Okay, that's a nice mechanistic story.
And importantly for validation, they show that the cells BPTES eliminated share characteristics with human senescent cells and look similar to the cells cleared in P16 DTR mice.
Those are the mice engineered specifically to allow senescent cell clearance, right?
Oh, yeah.
Kind of a gold standard comparison.
Exactly. So it suggests BPTES is hitting relevant senescent cell populations through a defined metabolic pathway, glutamine metabolism. That offers a potentially new, druggable target pathway for senolytics.
Sounds good, promising,
Yeah.
but there's always a but. What's the catch here?
Well, a couple of significant ones. First, BPTS itself, known to have pretty poor bioavailability. That's a major hurdle for actually using it as a drug in people.
Right. Tough to get it where it needs to go.
And second, the effect size. The senolytic effect they observed was only partial. They reported about a 30% reduction in senescent cells.
30%? How does that compare?
Well, compounds like ABT-263 or navitoclax often show clearance rates of 80% or even higher in similar models. So 30% is okay, but not amazing.
Not exactly wiping the slate clean.
Not at all. And the biggest caveat? Maybe. Absolutely no lifespan data.
So they cleared some senescent cells, but we have no idea if it actually made the mice live longer or healthier.
None whatsoever reported in this paper. And without that, you've got an interesting mechanism, maybe a tool compound, but not evidence for longevity intervention yet.
So, connecting the dots for biotech, what needs to happen next to make this finding stronger or more relevant?
Lifespan studies, period. You absolutely have to show that this intervention extends health span or lifespan in mice to validate its potential.
That's the benchmark.
It really is. Also, given the partial clearance, combination trials could be interesting.
Like BPTES plus another senolytic.
Yeah, maybe combine it with something like navitoclax or fisetin. See if you get synergistic effects, maybe hit different senescent cell populations or overcome resistance. Could it be part of a cocktail approach?
Okay. So where does this GLS-1 inhibitor fit in the grand scheme of senolytic development?
Well, it definitely adds to the evidence that targeting specific metabolic vulnerabilities of senescent cells is a valid strategy. We've seen that with other compounds targeting different pathways.
So it confirms the metabolic approach is viable.
Right. But this paper mainly provides nice single-cell resolution and details on the mechanism of this specific inhibitor. It doesn't really introduce a fundamentally new way to clear senescent cells, nor does it show breakthrough efficacy.
So again, valuable incremental progress, understanding a known pathway better.
Exactly. Deepening our understanding, but not changing the game this week.
All right, final paper for this week's review, moving into the brain. Functional connectome of superagers reveals early markers of resilience and vulnerability to Alzheimer's disease by Zhao et al.
This one's from Stanford University, also a bioRxiv preprint, posted Tuesday, July 22nd.
Superagers. Fascinating concept. People who maintain really sharp memory way into old age. What did Stanford find here?
They used functional MRI, fMRI, to look at brain connectivity. And they identified a specific pattern, a functional connectivity signature that they're calling the Alzheimer's Resilient Connectome, or ARC.
ARC, okay.
This signature, which involves connections mainly in prefrontal insular networks, was remarkably good at distinguishing superagers from age-matched individuals who already had Alzheimer's disease. High accuracy.
So it's like a brain fingerprint for cognitive resilience.
Kind of, yeah. And what's more compelling is that this ARC signature, even in cognitively unimpaired adults, predicted future outcomes.
How so?
People with a stronger ARC signature showed slower accumulation of amyloid beta plaques over time, about a 33% reduction. And they had a 40% lower risk of converting to mild cognitive impairment, MCI, over a 5-year period.
Wow, okay. So it's potentially predictive before symptoms really show up.
That's the exciting implication. They even found some potential genetic links genes called CLYBL and FRMD6 that seem to be associated with having either the superager ARC or an Alzheimer's-like connectome.
This sounds really promising for maybe early detection or risk assessment for Alzheimer's, but reality check time, what are the limitations?
Well, the practical one first. fMRI is powerful, but it's expensive, not widely available, not really practical for mass screening right now.
Okay, clinical utility is a way off. What about the science?
The bigger scientific caveat is, again, correlation versus causation. Does having this ARC signature cause resilience? Or is it simply a marker, a consequence of some other underlying biological factor that promotes resilience?
We don't know if the connectome itself is the protective thing or just reflects something else that's protective.
Precisely. We can identify this resilient brain pattern, but the study doesn't prove that the pattern itself is the key driver of that resilience.
So that raises the big question. Can we move from just identifying this ARC to actually enhancing it? Can we build resilience?
That's the million-dollar question, isn't it? The next logical step would be to explore things like neurostimulation techniques.
Like TMS, transcranial magnetic stimulation.
Exactly. Could you use TMS to target and potentially strengthen those prefrontal insular networks that make up the ARC? And would doing so actually improve cognitive resilience, maybe in preclinical Alzheimer's models?
Trying to actively build that resilient connectome.
That would be the key experiment to bridge the gap from correlation to potential intervention. Fascinating possibility.
So summing up this superager paper, what does it add to our understanding of cognitive aging and Alzheimer's?
It definitely builds nicely on earlier work identifying superagers, like the studies out of Northwestern. It provides much more detailed neurobiological insight into what makes their brains different functionally and even hints at genetics.
So better characterization of the phenotype.
Yes, a much deeper characterization. But it largely reinforces the existing idea that some people have brains that are inherently more resilient to age-related changes and Alzheimer's pathology. It doesn't propose a fundamentally new mechanism of Alzheimer's or a radically new way to intervene against the disease itself.
It helps us understand resilience better, but maybe not how to universally bestow it yet.
That's a good way to put it. It refines the picture of resilience, but doesn't offer a brand new intervention paradigm this week. So wrapping up our look at this week's papers. The overall assessment after digging into these four and screening the others is pretty consistent. We saw solid research, some really rigorous work, valuable incremental steps forward in AI diagnostics, epigenetics, analytics, neuroscience, all important areas. But it's clear that no single paper this week really presented what we call a model change breakthrough. Nothing that fundamentally shifts how we should be thinking about tackling aging.
And that distinction is so critical for researchers in this field, isn't it? Separating the impressive, necessary, incremental progress from the truly paradigm-shifting discoveries.
Absolutely. Most of these findings, as good as they are, end up reinforcing known paradigms. Senescence matters, metabolism matters, epigenetics matters, which is valuable confirmation and refinement.
But not the revolution we're always hoping for each week.
Not this week. To really move the needle towards, say, adding a decade or more of healthy lifespan, we're still looking towards and prioritizing those earlier, perhaps more foundational breakthroughs.
Things like partial reprogramming, maybe.
Partial reprogramming, maybe mitochondrial transplantation, things that seem to represent more fundamental shifts in how we might be able to intervene in the aging process itself. Those feel more like potential model changes.
So maybe a final thought for you, our listeners, to mull over until next time. What really are the signs of a true paradigm shift in longevity research? How do we get better at spotting them early and distinguishing them from this really impressive but ultimately incremental progress?
That's a great question to ponder.
Something to chew on. Thank you so much for joining us for this candid and hopefully critical look at the latest in longevity science. We really appreciate you being part of the Longevity Papers podcast.
And don't forget, you can always visit longevitypapers.com. We'll have the full list of papers we discussed today, plus links and other resources there.
We will be back next time for another look at the science. Until then, take care.